Hello and welcome to DNA Today. I'm your host, Keir Deneen. DNA Today informs you on what's happening in the genetic world. The show is packed with news stories and lessons, all to keep you updated and educated on the ever-changing world of genetics. And today on DNA Today, I'm delighted to share an interview with the Genetic Engineering Team here at UCONN. But before I do, I have to share a really exciting opportunity I had this morning. Dr. Charles Lee, the scientific director at JAX Genomic Medicine, Skyped into my genetics class today. So I want to share kind of who he is and what he discussed with the class today. So Dr. Charles Lee is a world-renowned scientist, best known for his discovery that copy number variation is a widespread and significant in the human genome. Now back up a little bit. What is copy number variation? Copy number variation is a state in which the cells have an abnormal number of copies of DNA sections, sometimes associated with susceptibility or resistance to disease. And different people have different numbers of these different amounts of these variations. And in his role as scientific director, Dr. Lee is responsible for the scientific direction and coordination of JAX Genomic Medicine in Farmington, Connecticut, not too far from UConn here. He earned his PhD in 1996 at the University of Alberta in Canada, and then held research fellowships at Cambridge University in England and Harvard Medical School. And after that, he joined the faculty in pathology at Harvard Medical School. He also introduced state-of-the-art clinical genetics tests as board-certified clinician cytogeneticist at Birmingham and Women's Hospital. And it's amazing. He's written more than 130 papers in top-tiered scientific journals and has held advisory roles for numerous national organizations, including the Food and Drug Administration, National Human Genome Research Institution, the American Society of Human Genetics, and the American College of Medical Genetics. So he has quite the resume, and it was very exciting for him to be able to Skype into my class. Thanks, shout out to Dr. John Malone for setting that up. And so today in class, he went over like kind of what Jackson is, and that it's 85 years old, the institution. And the headquarters, as I've mentioned multiple times in this show, is located in the beautiful, breathtaking Bar Harbor, Maine. And the largest supplier for mice for researcher is Jackson Laboratories. They have about 1 million mice breeding right now in Bar Harbor, Maine, their headquarters. And they have 3.5 million mice that are shipped out every year, which is just crazy numbers. And they really are the largest supplier. And some mice, it's interesting how they have different strains of mice. I actually was able to tour the headquarters up in Maine, and just hearing from them all the different types of mice that they have and how different types of mice are used for different types of research. And some mice are more humanized, and these are mice that can be tested to see which medications are effective for specific diseases. And this is something he gets into a little bit later that I'll talk about. But he was talking about how Jackson is focused on expansion, and the opening of the Jackson Laboratory for Genomic Medicine was a huge advance in this expansion. And this just happened a month ago in early October. And he's hoping, along with the other Jackson employees, that this will be groundbreaking work and it will cause a ripple effect in personalized genetic medicine. And I mentioned personalized genetic medicine a lot on this show, personalized medicine, and he mentioned this a lot, and that's really what Jackson Laboratories is all about, is really figuring out that personalized medicine and what medicine is going to be most effective for specific cases. I want to share 2 quotes with you. They're kind of linked, that he shared that I actually got verbatim, so I was happy about this. He said, "Right now, genetics is evolving at such a rapid pace and is infiltrating all aspects of medicine and healthcare." So genetics is at the heart of everything. It really is. When it comes down to it, genetics is at the base of everything. And Dr. Lee really emphasized this with what he was sharing with my class and his discussion. And it really just coming from him, it's very important to hear that it really is at the heart of everything, and understanding the genetics of things is square one. One main focus at Jackson Labs is cancer. And a statistic about cancer is that 1 in 2 or 1 in 3 people will develop cancer in their lifetime, which is a huge amount of people. Everybody knows someone that has had cancer, gotten through it, or died from cancer. And so it really is affecting everybody in some way. And Jackson Labs has a program where people with cancerous tumors that are being treated can have their tumor cells extracted and injected into specially produced mice that allow for this tumor to grow within them. Now, these mice are then tested with different drugs to see which is most effective for your personal tumor. And he really stressed that earlier diagnosis of cancer and more personalized medicine is the future of cancer treatment, and hopefully we're finding more and more things that can lead us there. Another big kind of area that he talked about Jackson being involved in was the microbiome and infectious diseases. And this is a very up-and-coming area that when I attended the Jackson conference, they really talked a lot about the microbiome and how it's kind of the next step. And this is really what a lot of research is being focused on. And George Weinstock is heading this microbiome initiative, and he spoke at the conference and talked about his own research. But Dr. Lee today shared that kind of what he does. And he works in the neonatal unit. This is George Weinstock. He works at the neonatal unit at Connecticut hospitals. And there he collects stool samples daily from babies and then sequences them to learn what bacteria is part of their microbiome. And with this information extracted from the samples, he can predict which babies will have an infection and then tell you, what antibiotic resistance those bacteria have in their system. And you really get an inside look on what's happening. And then because of all this information, medicine is so much easier to figure out what's going to work because you really know what bacteria are going on in there, what they're going to be resistant to. And so this is a very, very exciting field, and it's huge right now. And it's really exciting to hear that Jackson Labs is really at the forefront of this research. And I was lucky enough to ask Dr. Lee a question, and I asked him what the relationship between Jackson Laboratories and UCONN was. And he explained that Jackson isn't a hospital. It's an independent working site. And so it's very valuable to have a close affiliation with the hospital to work with physicians and get material from patients to really be able to expand their research and just having that close collaboration. And they have that close collaboration with UCONN because the Jackson Laboratory for Genomic Medicine in Farmington is on the UCONN Health Center campus. It's right there. Everything's integrated. They have their own building there, but they're right on site. So it was very exciting to hear from him, and I really want to thank Dr. John Malone for inviting him virtually into the class. That was an awesome experience, and thank you, Dr. Charles Lee, for coming into the class. Now, I want to share with you the interview I conducted last week with the genetic engineering team here at UCONN. The interview, we mentioned a meeting. I want you to pay attention to that. And I attended this meeting the day after the interview. And I have to say, they're a great group of people and are really energized about genetics. I like finding other people that are energized about genetics. And they really inspired me to get into research, more motivated. They meet Tuesday nights at 7 p.m. on the third floor of the Student Union. And so let's take a listen to what they had to say. Hello and welcome to DNA Today, where we explore new genetic advances. And today I'm joined by the genetic engineering team at UCONN. I'm here with Brian Lang, Jeffrey O'Brien, and Nick Genetti. How are you guys?
We're awesome.
Doing awesome. We're pumped up to be here. So, people are probably wondering, what is the genetic engineering team? So, does someone want to talk about, first say your name so they can get used to your voice, and then explain what your club does.
I'm Jeff O'Brien. I'm the president of the club, and our club is aiming to find out more things about synthetic biology, educate people in the UCONN community about synthetic biology, and also compete in a competition called the International Genetically Engineered Machine Competition, which is basically a collegiate competition where different colleges come together and present a synthetic biology project that gets judged.
Nice.
Hi, my name's Brian. I'm the secretary. And just a little bit more background on what iGEM is. iGEM started in 2005 as an outgrowth of an MIT activities program, and then in 2012 it became an independent organization that kind of runs its own thing now. And right now I think we have a little bit more international teams coming to national teams, so diversity is very awesome, and different educational backgrounds that people come from is also very varied, which is manifested in the projects that people do.
Hi, I'm Nick Genetti. I am a former iGEMer who now actually works at UCONN, so I'm kind of just around helping the guys with the club now.
You've got the perspective on things.
I have a perspective, yes.
Nice. So what are your individual backgrounds when it comes to education experience and particularly lab experience?
Well, on the UCONN campus, I'm in a lab, the Gogarten lab, which is a bioinformatics-based lab, and basically it studies molecular evolution.
Nice. And so are you an undergrad?
I am an undergrad. I'm a senior.
Nice. And what are you majoring in?
Molecular cell biology.
Okay.
I'm a sophomore, and I'm majoring in the same major as Jeff, molecular cell biology. And in terms of lab experience, I've been doing research during the summer since my sophomore year in high school. I've been doing research in various areas such as EEB, pharmacology, and pharmacology again. And yeah, I've had a lot of experience with regular just pipetting, and at this point, I haven't had much experience with genetic engineering techniques such as transferring vectors and putting DNA in different organisms. But as part of this club, we will be teaching people, including myself now, how to do these methods.
So it really is a teaching environment. It's not necessarily you go there and you really have to know what you're doing. You can kind of start from square one.
Neither of us really have that experience in genetic engineering techniques per se yet. Most of our experience is more towards just molecular cell biology in general. Like my lab actually doesn't do any work in wet lab. We do all computer-based work, but we're brought to this club by a common interest in that we want to start doing these kinds of things at UConn because there aren't too many opportunities at the time. And we're hoping that this club will bring those kinds of opportunities to undergrads.
Right.
And Nick, as a former member of this, can you explain kind of what your experience was before and now after of where you are today?
Sure. So I am a UConn alum. I graduated with a bachelor's in biomedical engineering and a master's in molecular biology. And I actually competed on the 2012 iGEM team here at UConn, where, should I say the project? I don't know. We attempted to create a yeast organism that was capable of synthesizing vitamin D. The idea, apparently, we had a lot of drinkers, not encouraging that, on the team that year, and we wanted to create a healthy beer for people. So we did actually get to do, I want to say like 60% of the project. We ran out of time at the end, and we were able to create what's called a shuttle vector, which put the genes we needed into E. coli, and we just never managed to transform it into yeast. So we got almost there.
But you were successful with the E. coli of actually getting the genes in there and they were expressing the protein?
That was another thing we never got to.
Okay.
We, like I said, we ran out of time. It was...
But you got to explore the concepts and really
got,
get experience.
Yeah, a couple of the members that year definitely got into the lab. They were able to create the shuttle vector, which, like I said, is just a vector that has both yeast and E. coli or points of origin that can express in both organisms. And we definitely got it to express the genes because we confirmed that with PCR, but we never actually got to do like a mass spec or something to make sure the proteins were being expressed.
Mm-hmm.
So.
And what are you doing now?
Now I am a lab tech in Rachel O'Neill's lab, Dr. Rachel O'Neill here on campus. Who
was interviewed 2 weeks ago.
Mm-hmm. Yeah, so you guys probably all know about her research, so I participate in that and also the chromosome core, which is a karyotyping and cytogenetics institute, I'll say, for lack of a better term, here on campus.
Nice. So you mentioned that, you've been, Nick, to the iGEM competition, but also you guys just came back from the annual competition. Can you express, the excitement there, what was going on, and just kind of put us there?
Yeah, we went there as volunteers this year, and it was definitely an exciting time. Just seeing all those groups do projects that they were interested in, it definitely makes a difference when there's a student drive to do a project like that, from what I've noticed.
Yeah, when I was there, it was just magical. I just didn't want to leave because, first off, different people from different countries speaking different languages. Internationalism was huge there. Like, I found myself standing in line for lunch, and I couldn't understand anybody because one person was speaking German, another group was speaking Norwegian, and I think another group was speaking Japanese or something, and it was just great.
And where was the competition held?
It was in Boston, Massachusetts.
Oh, nice. So not too far from us here at UConn.
Exactly. We just drove up or bussed up.
They're actually planning on having it there again next year, I believe.
Oh, really?
Mm-hmm. Yeah.
Oh, okay, sweet.
Nice. And you guys are currently working on your project to be entered in the competition next year, correct?
Right. After coming back from the competition, the excitement kind of springboarded us to really wanting to at least get some kind of project idea going so we could start for next year.
Definitely. So what are, do you remember any specific projects there that you saw, or can you share with our listeners, the type of projects you saw and maybe who did them and all of that?
Well, there was this one that I really liked the idea from. and especially the fact of how complete their project is. Because for one part, a lot of the projects tend not to be complete, as you heard from our previous competition, but this one had a lot of supporting data. UC Davis did a project where they were able to identify whether or not olive oil is rancid. Apparently, in the U.S. and other countries too, a lot of the olive oil that you find on the shelves is completely rancid. If it doesn't have if it doesn't have an expiration date on it, the company pretty much knows that it's rancid and they don't care. And so they developed a method to find out whether or not this was rancid. They used 3 different genes that were able to test whether or not certain compounds in the olive oil were broken down. I think they were testing for oxidation of longer chain fatty acids, which is indicative of olive oil being rancid. Basically, there's a certain profile of which fatty acids would be indicative of how rancid the olive oil is. So they used 3 different enzymes to test for these and made a small machine that was able to take a drop of the olive oil and see whether or not it was rancid.
Wow, that's awesome. That's really cool that they were able to actually take the project from the beginning all the way to really finishing it. As you said, a lot of projects are kind of a work in progress. What are you guys currently working on? Can we know, or is it top secret?
Well, we've been throwing around a few ideas. One of our members brought one up last week that I think both Brian and I liked, but I don't know if he wants us to talk about it or not.
All right, we can understand
it has
that.
to do with his research on campus, which he hasn't started yet.
Oh, you're right.
Yeah.
Yeah, we're always looking for new ideas, though.
That's
so
true. We try to bring it up at every meeting.
Of like what you want to do, like just starting from square one of like, okay, like where should we go from here? Like what should we start with?
Right.
Yeah.
We usually try to identify a problem first, which is the basic like engineering framework. Identify a problem and then propose solutions for it using the genetic engineering or synthetic biology framework.
And one of the things that iGEM has taught us is that it's not all about competing against each other, emphasis on against, to see who can come up with a better way to solve the same problem. Sometimes, like for example, the team that won the grand prize this year from the University of Heidelberg, Germany, they literally just found a solution to make genetic engineering more advanced. Like there wasn't a specific solution that was like harming humans like drought or starvation or something like that. It was just about improving the field of genetic engineering in general.
Yeah, and that's kind of what this competition is all about, is just getting students more involved in this basically a newer scientific field, and basically all these students coming together and learning about it together.
Interesting. So how do you begin to like modify an organism? Where do you start? Are there specific techniques and methods you use or technology, or does it really differ on the project?
So that's part of what iGEM and another foundation called the BioBricks Foundation is all about. They're working on creating standard biological parts that you could apply to a number of different problems and scenarios. So the idea being that you look at biology almost as if it's a machine. You take a gene and look at it as a part that can be interchanged between different organisms and basically apply to different problems. Like one technique that I thought was interesting was a couple years ago, a team, I'm sorry, whoever this is, I don't remember who it was, they created a gene that was activated by blue light. And another team created a gene that would allow a bacteria to produce a massive amount of cellulose. And at the competition, there was a visiting artist who took both of these concepts and combined them into be able to grow her own clothing. So she would create a vat of basically E. coli, I think it was E. coli, and almost like a 3D printer that she would shine light on the shape of the fabric that she wanted, and it would grow on a layer, and then the bottom would drop a little, and it would grow another layer, and then she would sew them together and create clothing, basically. So,
wow, that sounds like science fiction to me. It doesn't even sound real. It's so advanced.
Some of the things that you see at that competition
it was definitely
are a lot like science fiction.
Yeah, so the idea is that modifying an organism is going to, in the future, become almost commonplace, where you can take different parts and fit your needs. And I should mention that a big part of iGEM is safety. So there are a lot of teams that design sort of biological safety switches, so
Kill
to speak.
switches.
Kill switches.
Mm-hmm.
Yeah. So the idea being that what you're designing will stay exactly where you want it to be, and it won't leak out.
It's not going to be out of control.
Yes, exactly.
Interesting. So how has this experience helped you with your own personal career goals? I know, Jeff and Brian, you're still in school. You're both undergrads. And Nick, you're moving on to bigger and better things. So how has iGEM really kind of festered your career goals and gotten you to where you're working towards?
Well, I guess it's a little bit different than the lab experience that I've had on campus, which mostly in lab experience I'm working with PIs or mainly graduate students, not as many undergraduates. But this has given me the opportunity to really connect with people who are also undergraduates and interested in science, which tends to have a bit of a different cooperative feeling to it. And I think that's definitely important in science, is cooperating with other people who are interested in what you're doing.
And I would second that, because most of the time when I've worked in labs, it's usually me trying to spearhead my own little thing with the help of my mentor, or me just following along with my mentor. But now that it's student-driven, or should be student-driven, we have to really think for ourselves and really work together. Even though there may be some conflict, we have to learn how to work through it, regardless of what we may or may not like about it. So that's just this kind of idea of collaboration with peers is something that will probably help me when I become a doctor, because I'll have to collaborate with nurses and other doctors and discuss with all of them and just keep on moving on.
Interacting on a professional level.
Yeah, exactly.
Definitely.
Well, I can speak for a couple of the guys that graduated with me who did the iGEM back in 2012. They said that it really helped them on interviews, because a lot of people have never heard of iGEM before, so you see a little thing on, oh, what's this? And then they explain what they did, and it's just a talking point during interviews about, like, oh my God, you did this during your undergrad. That's awesome.
It all sounds very impressive in what you're doing, and really just kind of getting your feet wet with all of this. Definitely. So if people want to join, what do they need? A certain background, or are you really open to a lot of different people?
Frankly, anybody can join. For example, I sat down to eat lunch with the Cornell team while I was up there at iGEM in Boston, and they had a psychologist on their team. He just focused on the human applications and the human policies that can be used with their project. Even though he didn't necessarily have a background, he certainly made good contributions to the team. So basically anybody can join. Engineering, social sciences, hard sciences, whatever, anybody.
Right. A lot of the projects, they're multifaceted. They're not just focused on the project themselves, but also expanding the knowledge of synthetic biology to the public. So they like to see when projects will incorporate the public and public education on these fields, especially because there does tend to be a lot of fear about synthetic biology, and that's probably stems from lack of knowledge basically for laymen's. So if you aim to basically expand the knowledge in the public, it helps your project out. The judges will look favorably on that.
Definitely. So you like incorporating a lot of different people with different backgrounds into this, like you mentioned, psychology major for a different team of having different experiences and all coming together so that you're not all the same type of mind. You kind of can collaborate a little bit more.
Right.
Exactly. You don't need to be a science major to do community outreach.
Yeah,
Definitely.
And iGEM itself has different facets to the competition, where a lot of people just focus on the wet lab aspect, which is the actual creating or modifying the organism. There's also a software component to the competition. There's an entrepreneurship component to the competition, in which it would be bringing the project into a company or something along those sorts. So there's a lot for people to get involved in.
And as you said, there was the clothing, the lady who had the kind of business of doing that. So they took this idea and this project and kind of ran with it and turned it into a business.
Well, she was more for art, but
there
are several businesses that are cropping up around synthetic biology and genetic engineering. So I know I read a year or two ago, there's a group in Spain that's using a modified algae to produce hydrogen. I think the UC Davis team you were talking about is looking to get FDA approval for their technique. Mm-hmm. So, I mean, it's branching out and becoming really the way that society is going to be moving.
And it's a great starting point for undergrads and even grads.
Oh, yeah.
Right.
Definitely for undergrads.
Not all of the projects at iGEM will go on to become companies, but there definitely are a few that, when their project is very complete, they'll go on and start a company based off of that.
Great. So when does your team meet? How can people get involved?
So we meet on Tuesdays from 7 to 8 in the student union room of 322. Well, for tomorrow we're meeting in 312, but for the rest of the year until December, we're meeting in 322, 7 to 8.
Yeah, that time will definitely is flexible based on the schedules of people who want to join, and we're definitely always open to new members. Anyone who wants to join could either just show up at a meeting or
email one of us.
Email one of us, which I think we're both listed on the UCONN page for Genetic Engineering Team.
Do you want to just share your emails just in case people can't find it?
My email is jeffrey.o'brien@uconn.edu. So that's jeffrey.obrien@uconn.edu.
And my email is Brian with an I .liang, so Liang @uconn.edu.
Great, and we'll include those in the show notes so you can also find it there. That way we really resaturate them with that information. So that's great. So I want to end the interview here with asking you how you got inspired in genetics. I got inspired back in high school and really pursuing it myself. But I want to know what captures you for genetics.
Yeah, I think it was also in high school for me. I took a AP biology class in high school, and basically that was the point where I realized that I wanted to study biology in college and go on to med school, which is my eventual goal. And I think what's really fascinating about genetics for me is just how it relates all organisms. I mean, like I said, my lab that I work in here on campus studies molecular evolution, and just knowing this common theme that ties together all organisms is just a really fascinating.
The DNA is so universal.
Right.
The thing that inspired me to go into genetic engineering team was Jeff. Thanks, Jeff. I first met him at the Honors Activities Involvement Fair, and he told me what it was about, and I got interested in it. And then what really pushed me to stick with this club was when I went to iGEM just last week, because I got to see what the possibilities could be with genetics. Like, you could do stuff with art, you could do stuff with health, you could do stuff with food, you could do stuff with entrepreneurship, like all of these things just with genetics. Amazing.
Definitely. How about you, Nick?
So mine interest in genetics started in high school from a couple of great high school teachers. And one of them in particular, I was taking a pre-engineering course at the time and biology, and they're like, what do you want? Can you like both of these subjects? Why don't you find a way to combine them? So I've always been looking for sort of the genetic engineering synthetic biology outlet, and then I kind of just ran with it in college. So
Definitely. Well, thanks so much for sharing. You guys, listeners, can find information on them. We mentioned their emails, and obviously we've talked a lot about their club here today on the show. But you can go to UContact. Am I saying that right?
Yeah,
UContact.
right.
Okay,
Right.
Yeah, and you can just Google them. Google's a great source. It pops up.
We might still be under International Genetically Engineered Machine Team on UConn.
So iGEM.
Yeah.
Mm-hmm. Yeah, so you can find them on there. And I definitely encourage you to join. I'm definitely going to go to a meeting. I do have to say that. I happen to be free during that time. So I definitely want to come by and hear what it's all about. And I'm sure I'll be talking about them much more on future episodes. So thank you so much, guys, for coming on. Really appreciate it.
Thank you for
Thank
having
you.
us.
That was really cool just to hear from them. And it doesn't get much cooler than that. They're modifying organisms to help society to solve a social problem, a problem that's facing us today. So pretty cool. Well, that about wraps up the show for today, for DNA Today. Tune in next week, Monday at 1 p.m. and every Monday at 1 p.m. to learn more about genetics. You can go to whus.org to listen. And you can also check out the website, dnapodcast.com, for all kinds of content on there. And everything that I've shared on the air here can be found on dnapodcast.com. If you're interested, follow on Twitter at DNA Podcast. Follow me on Twitter at KIRADINEEEN. That's at Kira Deneen. And if you have any questions you want answered on the air, shoot me an email at info@dnapodcast.com. Thanks for listening, and join me next week, Monday at 1 p.m. to learn and discover new advances in the world of genetics.
listening to Health Chats, WHUS, with Shilpa and Yasmin, every other Monday from 1:30 to 2 p.m.
Hello everyone, this is Yasmin and Shilpa with Health Chats on WHUS Stores at 91.7. As the end of the semester